in this topic, and then we can move on to the lecture questions. So the first thing we're going to talk about is the accuracy of DNA replication. The big point is that DNA replication is very accurate. So when you look at it, you can get mistakes during DNA replication. And if you look at just one subunit of DNA polymerase III, and this is getting to one reason that it actually is so accurate, when you just look at one of the subunits of DNA polymerase III outside of a cell, so that's what it means in vitro, it actually will introduce an incorrect base in every 10 to the 4th bases. So that's 1 in 10 to the 4th. But when you actually measure the mutation, so basically if you had one mistake happening every 10 to the 4th bases, then you're going to expect that mutation rate, at least that mutation rate in bacterial cells, because if you incorporate an incorrect base, that's going to change the DNA sequence, and that's a mutation. And so when you actually measure the mutation rate in bacterial cells, it's much less than that. You only get one mistake in 10 to the 9th nucleotides, which means that something is fixing all those mistakes that are being made with the alpha subunit of DNA polymerase III. Most of your increased accuracy comes from the fact that DNA polymerase III or DNA polymerases have a proofreading function to them. So they're not only connecting together nucleotides, they're also checking to make sure that they added in the correct nucleotide. And so what they can do is if they discover that they've added in the wrong nucleotide, they can back up, excise it, and then replace it with a new nucleotide and then move on. Okay, so a lot of the increased accuracy that you see in vivo is due to this proofreading function that DNA polymerases have. There's also DNA repair pathways. So I took that slide out. There's also DNA repair pathways in the cell that will further increase the accuracy of DNA replication. So if there's a mistake that's made that then doesn't get fixed by DNA polymerase, there is still the opportunity for these repair pathways to fix that DNA damage. Okay, so the last thing that we're going to talk about in this topic is how the ends of linear chromosomes are replicated. And the reason that we have to talk about this, and we're not going to get into the details of it, but suffice it to say that when you replicate a linear chromosome, you're going to lose a little bit of DNA at the ends. Because of the way that DNA is replicated, one of the strands you're not going to be able to synthesize all the way to the end, and so you're going to lose a little bit of DNA every time you replicate the DNA. So what that means is every time the cell divides and you need to replicate the DNA, you're going to lose a little bit of DNA at the ends of the chromosomes. Okay, so and the ends of linear chromosomes are called telomeres. Okay, so the telomere is the end of a eukaryotic chromosome, and so each end is a telomere. So you have 2 telomeres per chromosome, and a telomere is simply a short sequence that gets repeated over and over, and then you have this thing called a 3' overhang. Okay, so for example, this is GGG-TTA. That's the short sequence, and then it just gets repeated GGG-TTA, GGG-TTA. So it just gets repeated over and over again, and that's what the telomere consists of. And telomeres can consist of that one repetitive sequence 100 or 1,000 times. Sorry, it repeats that sequence 100 to 1,000 times. This is showing you the sequence that gets repeated depends on the species. So this is the human telomere. And this can be repeated 100 to 1,000 times at the ends of a linear chromosome. And that's simply what the telomere is. Okay. There's also proteins that are complex with the telomere that we're not going to get into. And then we're also not going to talk too much about the 3' overhang, but that's the part of the telomere where it's single-stranded and it doesn't have a complementary part to it. Okay, so every time you replicate the DNA, it's going to become shorter. And so one question that you might ask is, well, what is going to happen in terms of chromosomes that you pass on to your offspring? Do they just, if that happened and they became shorter, eventually there'd be nothing to pass on. In your germ cells, you have this enzyme active or being expressed. It's called telomerase. And the function of telomerase is that it lengthens the telomeres. Okay, and this enzyme is active in your germ cells where it lengthens your telomeres, and that's important because then you're not passing on shortened chromosomes to your offspring. You're passing on chromosomes that have the proper length. Most of your somatic cells, so most of the cells that make up your body, do not have this enzyme active. And so their telomeres will become shorter every time they divide. Okay? And one thing I told you in terms of cancer that cancer cells have to do is that they either have to activate this enzyme telomerase because they can divide indefinitely, or you consider them immortal. And so in order for them to be able to do that, they're going to have to find some way of stabilizing their telomeres. And they can either do that by expressing telomerase or finding some other method of stabilizing the telomeres so that they don't become shorter as you replicate the DNA. And so, again, this is going to be the telomerase is the enzyme that lengthens the telomeres. You can also think of the telomeres, there's no genes within the telomeres, so in a normal cell when they shorten, you're not getting rid of any genes when they shorten. And what happens in cells is if you take a cell out of the body and put it in a tissue culture dish, that cell can only divide a finite number of times. So there's only a certain number of cell divisions that it can undergo, and then it stops. The cell doesn't die, it just stops dividing. And that point at which it stops dividing is correlated with the shortening of the telomeres. So the telomeres become so short and then the cell stops dividing. And there's a correlation between the shortening of the telomeres and the lifespan or the number of divisions that a cell can undergo. And so that's why cancer cells, if they want to divide immortally past that number of divisions, they're going to have to either express telomerase or find some other way of stabilizing their telomeres. Again, this enzyme is active in your germ cells, but not active in most of your somatic cells. Okay, so that ends DNA replication. So we're going to do the lecture questions for this topic. And so the first question that I want to ask has to do with one of the enzymes. So what enzyme builds the new strand of DNA during DNA replication? Right, DNA polymerase. And one thing we stated about this polymerase is that it requires a primer. First of all, what is a primer? Yeah, so in DNA replication, it's a short strand of RNA. And so why is it needed then? Right, so it can't start a new strand on its own. It can only build off a pre-existing strand. So the primer is going to be the pre-existing strand that's laid down that the enzyme can build off of. And then what enzyme lays down this primer? Right, DNA primase. So number 5, describe the roles of DNA helicase. So what does that do? Right, separates the two strands. And then what do the single-stranded binding proteins do? They keep it separated, right? So they bind to the single-stranded regions and keep it separated. Number 11, where does the energy for building the new strand of DNA come from? Right. Started off right.
Ribose, deoxynucleoside triphosphates?
Right, deoxyribonucleoside triphosphates. And so that's the D-ATP, D-CTP, D-GTP, and D-TTP. We said the energy for synthesizing the new daughter strand comes from those nucleotides. Okay, so number 18, this gets to how DNA is replicated. So we said DNA is replicated in what type of manner? Right, C, semi-conservative. 20, which of the following statements best describes DNA polymerase? Right, so That kind of incorporates the two things that I told you about DNA polymerase. One, that it can't start a new strand on its own, so it does require the presence of a short primer. And then it can only build onto the 3' end of a pre-existing strand. And so it always builds in the 5' to 3' direction. So that's the one that most accurately describes DNA polymerase, that it can only build off a primer, and that it specifically can only add onto the 3' end of a growing strand that it's building. 21, this enzyme catalyzes covalent bond formation between fragments of DNA. Right, B ligase. 22, blank produces short sequences of RNA, which allows DNA polymerase to synthesize a new strand of DNA. Right, that's DNA primase. So the short sequence of RNA that they're referring to there, that's the actual primer. Okay. Number 24, why is there a need to... Okay, so Okazaki fragments. So I told you that the lagging strand you have to build in fragments. The name for those fragments, they're called Okazaki fragments, and that just comes from the guy who discovered them. His name was Okazaki. And so they're basically asking you, why is there a need to produce one strand in a series of fragments versus the other strand as a one continuous strand? So why do you have to build them differently is what it's asking. Right. So it's a combination of two things. So one, because the two strands are anti-parallel to each other, and then also because DNA polymerase can only build in one direction, you're going to have to build the two strands differently from each other. Okay, so that's the last one that I wanted to do. Do you guys see any other questions in these lecture questions that you want to do?
So, like, what question, like, I don't know what's on there, but, so, like, the primers, is it that the primers are only built in the 5' to 3' direction, and that DNA polymerase can only, like, I don't know, start from there, or is it that...
They can only build, both of them can only build in the 5' to 3' direction. And then primase can start a new strand, and it lays down a short strand of RNA, and polymerase cannot start a new strand on its own, but it still synthesizes in the 5' to 3' direction. It just so it always builds off the 3' end.
Okay. I wasn't sure, like, what caused
I don't know the cause of it either, so yeah, I can't answer that question for you. Yeah, it's just how the enzymes work. So it does have something to do with the enzymes themselves in terms of how they work as to why they do that. Other questions? Okay, so let's move on to the next topic, which is gene expression. And essentially what we're talking about in gene expression is how does a segment of DNA code for a protein. And so in gene expression, we're talking about how does a cell take a segment of DNA called a gene and using that segment of DNA end up producing a protein. Okay, so when we say a gene is expressed, what we mean is that the cell is making the protein that that gene codes for. Okay, so if a gene is expressed, that means you're making the protein that the gene codes for. And if a gene is turned off, or we say that it's not expressed, that means that you're not making the protein that that gene codes for. And so we're going to talk about gene expression first, and then after gene expression, we're going to talk about how you regulate that process, so what determines whether it's off or on, meaning what determines whether it's going to be expressed or not expressed. Okay, so in order to express a gene, you're going to have to go through two processes, transcription and then translation. And so this is diagramming out a relationship that you've already seen in the course. And so essentially the gist of what happens in transcription, you're going to use DNA as a template to synthesize an RNA molecule. And then in translation, you're going to use RNA as a template to produce a protein. And obviously we're going to go through the different enzymes that are needed in these processes and then the details of how they occur. Also, just to say it in a slightly different way, what's going to happen during transcription is the sequence of nucleotides in one of the strands of DNA is going to determine the sequence of nucleotides in the strand of RNA that you build. And then in translation, we're going to see how the sequence of nucleotides in an RNA molecule is used to determine the sequence of amino acids in the protein that you're producing. Okay. And so for this class, when we talk about a gene, we're going to use the definition that a gene is a segment of DNA that codes for a protein. Now this isn't not accurate, but is a segment of DNA that codes for a protein, because there are genes that only code for an RNA molecule and don't ultimately code for a protein. So they only code for an RNA molecule, and then so basically what would happen is you would transcribe that gene to produce the RNA molecule, but then that RNA molecule is not used in translation to produce a protein. It's used for a different function inside the cell. So there are genes that only code for RNA, but then all the genes that code for proteins are going to have to go through both these processes to produce the protein. So you never go from the gene to a protein in one step. You always have to go through transcription and then translation to produce a protein in the cell. Okay, so the first thing we're going to do is just compare gene expression in a bacterial cell versus what has to happen typically in a eukaryotic cell. So this is just breaking up the two processes. So this is showing you a bacterial cell. In the first box you want to put transcription, and in the second box you simply want to put translation. Okay, so the first box is transcription, the second box is translation. And what this picture highlights is that those two processes in this cell happen in the same area. So they both happen in the same area. And what that means is that we're going to see that in bacterial cells, ribosomes can start to translate, you can start to make a protein before you're done transcribing. So meaning translation can start before transcription is finished, and that's one reason that that can happen in bacterial cells is because the two processes happen in the same area. Now when you compare that to a eukaryotic cell, there's going to be a couple of steps between transcription and translation, and then also transcription and translation do not happen in the same area. So in this first box, that's going to be transcription again. And then the second box below that, that's going to be RNA processing. And then the third box over here to the right, that is export of the RNA out of the nucleus. And then the very last box again is going to be translation. Okay, so you can see that in a eukaryotic cell, transcription happens in the nucleus and translation happens out in the cytoplasm. So that's one reason why in eukaryotic cells, transcription has to finish first before translation can start. So you don't have the two, you don't have an RNA molecule that's not done being made and then it's already being translated. That doesn't happen in eukaryotic cells. Another reason that doesn't happen is you also have these this additional step of RNA processing. And so the RNA that you actually produce is not the same as the RNA molecule that's going to be translated, and we'll see how it's different. And that process also has to take place before the RNA molecule can be translated. Okay, so two key things that you want to get from comparing those two slides is that in a bacterial cell, transcription and translation happen in the same area, which would be the cytoplasm. And then in eukaryotic cells, transcription and translation happen in separate areas. Transcription happens in the nucleus. Translation happens in the cytoplasm. And then in addition to that, for gene expression in a eukaryotic cell, you have to also go through RNA processing and export of the RNA out of the nucleus in terms of producing the protein in a eukaryotic cell. So you have those additional steps that don't take place
typically in a bacterial cell. Okay. Also, one thing that you can get from the slide that I'll mention when we go through it is that RNA processing also happens in the nucleus. So the process of RNA processing, that happens also in the nucleus. Okay, and so what we're going to do is watch this animation, which just basically goes through simple gene expression, just explaining transcription, and then translation. And genes that code, I think they use it in this animation, they might use the word structural gene. That's the name for genes that code for proteins.
The sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of a specific protein gives the cell and the organism the specific trait coded by the gene.
No. That's why I said like the definition we're using isn't totally accurate because there are genes that do not code for. They don't have a special name, but there are genes that. So, for example, we said that there's different types of RNA. So rRNA and tRNA, those are coded for by genes, but you don't use rRNA and tRNA as a template to make a protein. The only RNA that you use to make a protein is mRNA. And so the genes that we're talking about code for an mRNA molecule, and then the mRNA molecule is used as a template to make a protein. Why didn't they just what?
They tell you to like say is it a similar DNA that codes for RNA? You can't be wrong.
Because mostly what we talk about in this class is protein expression, and so it's easier that way. Okay. And we don't spend a lot of time talking about RNA molecules that don't code for proteins. We do talk about tRNA and rRNA, and we will talk about these RNA molecules called microRNAs and siRNAs and snRNAs, but we don't go into a lot of detail in terms of how they function. Okay, so the first thing we're going to obviously go through is transcription. And so we're going to start by first watching this animation that describes transcription, and then we'll obviously go through the process together.
A structural gene is made up of a sequence of bases in a DNA molecule consisting of a coding region with an upstream promoter and a terminator downstream of the coding region. The transcription of a gene into a messenger RNA sequence is initiated when RNA polymerase attaches to the promoter region and forms an open complex. RNA nucleotides are then polymerized from nucleotide triphosphates to make a messenger RNA. The messenger RNA sequence is elongated as the RNA polymerase moves down the DNA molecule until the RNA polymerase reaches the terminator region. Transcription is terminated when sequences in the terminator region are encountered. This causes RNA polymerase to dissociate from the DNA molecule, and the completed transcript is released.
Okay, so that kind of describes essentially what happens in transcription. We're going to go into a little bit more detail as we discuss it. So one of the first things that you want to realize in transcription is what is the product. So again, the product is an RNA molecule. So the product of transcription is going to be an RNA molecule. And so in this case, you're going to be using RNA nucleotides when you synthesize an RNA molecule.
So the nucleotides that are used in transcription are going to be ATP, TTP, GTP, and CTP. And those are the same as the nucleotides that are used in DNA. Obviously, oh, sorry, I made a mistake. UTP, not TTP, because you use uracil instead of thymine. But the difference one of the differences between these nucleotides and the nucleotides that are used in DNA is the pentose sugar. So all these nucleotides have ribose as their pentose sugar, and then the A, U, G, C tells you which nitrogenous base that nucleotide has. So A is adenine again, U is uracil, G is guanine, and C is cytosine. And so you're going to be using these nucleotides when you synthesize a strand of RNA, which is what you're trying to make. And again, in this process, you're going to use a strand of DNA as the template, but you're only using one of the strands. And whatever strand is the template strand, it's always the template strand for that gene. Okay, so it doesn't switch between the two strands. One strand is designated as the template strand, and it's always that strand. So I'm just going to draw out one strand of DNA. So here's one strand of DNA. And then this strand we'll say is the DNA template strand. And what I'm not drawing, obviously, is the complementary strand that would be a part of that DNA double helix. And then that's going to be used as a template. And again, for this process, it's the same, sorry, it's not the exact same, but you're using the base pairing rules again. So this serves as a template because if there's an A here, then RNA polymerase is going to come in and start with a U because U pairs with A. And then because there's a T here, RNA polymerase is going to connect a nucleotide that has adenine to this nucleotide. And then it's going to put a C, a G, a U, a C, a G, and then another U. And so again, the sequence of nucleotides in this template DNA strand is determining the sequence of nucleotides in the RNA molecule that you're making. Okay. Okay, so that's how DNA is going to serve as a template for the synthesis of the new RNA molecule. And again, you're just using the base pairing rules. Okay, so this repeats one thing. The first bullet point repeats something I've already told you. So only one of the two strands of DNA serves as a template during transcription. They mentioned in the animation that the enzyme that's going to build the RNA molecule is RNA polymerase. And so this is the polymerase that connects together these nucleotides to form the strand of RNA that you're making. And then I also told you that the nucleotides that are going to be the building blocks for making the RNA molecule are going to be ATP, CTP, UTP, and GTP. And then just to contrast this with DNA replication, RNA polymerase can start a new strand on its own, so no primer is required in this process. So RNA polymerase can come in, start a new strand on its own, so you don't need some type of enzyme that lays down something that would be equivalent to a primer for this process. Okay. Also, when you replicate DNA, you're replicating the whole chromosome. But when you do transcription, you're not transcribing the whole chromosome. You're not using the whole chromosome as a template to make this seriously long RNA molecule. You're using only a segment of the chromosome, and that segment is a gene, and you're only going to transcribe that very small segment of the chromosome. And so you have to have a segment of DNA that tells you where the gene begins, and then you have to have a segment of DNA that tells you where the gene ends.
And like for the template, I mean, does it automatically know which side to read?
No. So one thing that's involved in determining that is the promoter, which is what we're getting into now. So in bacteria and then also in eukaryotic organisms, the segment of DNA that tells you where to begin is called the promoter. So that's why it says it signals the beginning of transcription. And what the promoter does is it binds RNA polymerase. So the enzyme that needs to synthesize the new RNA molecule is first going to bind the promoter and start there. Then it's going to move and transcribe the region between the promoter. And then in bacteria, the segment of DNA that tells you where to stop is called the terminator, which is, hopefully you can remember that. Terminator is where you stop. It's given a different name in eukaryotic organisms. You don't need to know it for eukaryotic. It's not called a terminator, but it performs the same function in eukaryotic cells. But it's always a promoter regardless of prokaryote or eukaryote, and then the terminator is only prokaryote. So the promoter tells you where to start, the terminator tells you where to end. So essentially what's going to happen in this process is RNA polymerase binds to the promoter, transcribes the region between the promoter and the terminator. Then once the RNA polymerase transcribes through the terminator, it unbinds from the DNA, so it releases the DNA, and then it also releases the RNA molecule that it was synthesizing when it transcribed that region. Okay, so that's the gist of what's going to happen during this process. And so just a couple more things before we get into the details of what happens. So one thing that you just asked is how do you determine which one's going to be the template strand? And that's decided by the promoter. So the orientation of the promoter is going to determine whether the top strand or the bottom strand serves as the template. And it depends on the gene. So the only reason I have this picture down here is that this is the E. coli chromosome. And so you can see that for some genes, the top strand is the template strand, but for other genes, it's going to be the bottom strand. So it depends on which gene you're talking about in terms of what strand is going to serve as the template strand. Okay, but it's always, for whatever gene, so for gene A, it's the top strand here that's the template. For gene A, it's always the top strand. For gene B, it's the bottom strand that's going to be the template, and it's always the bottom strand. So for whatever gene you're talking about, it's always the same strand, but again, the promoter is going to determine which one, or basically think about it this way, the promoter is how the cell knows which one is supposed to be the template strand. Okay. So we're going to split, let me see what I put next here. Okay. We're going to split transcription up into 3 stages. And what we're going to start with is talking about what happens regardless of what you're regardless of whether you're in a prokaryotic cell or a eukaryotic cell. We're going to talk about what happens during each of the stages. And so we're going to start off with the initiation stage. The first thing that has to happen in the initiation stage is RNA polymerase binds to the promoter. So this has to happen first. And then the DNA strands unwind. You don't completely unwind them, just in the region where you're transcribing. So DNA strands unwind, separate from it. Oh, we'll say separate. DNA strands separate from each other. And then RNA polymerase initiates RNA synthesis. Okay, and then that's the things that you need to remember in terms of what generally happens in the initiation stage. Okay, and then the next stage is the elongation stage. And so during the elongation stage, RNA polymerase reads, and I'm going to put that in parentheses, RNA polymerase reads the template DNA strand in the 3' to 5' direction. So what that means is it starts at the 3' end of the template strand and moves along the template strand towards the 5' end. Okay, and so while it's reading in the 3' to 5' direction, it's it's producing the RNA molecule in the 5' to 3' direction. So RNA polymerase reads the template in the 3' to 5' direction and produces the RNA molecule in the 5' to 3' direction. So RNA polymerase, like DNA polymerase, can only add on to a 3' end. What did I do here? Direction. Okay, and then in the termination stage, the RNA molecule is released. And then RNA polymerase detaches from the DNA. And then we said for bacteria, this is going to happen when RNA polymerase goes through the terminator. Okay, so I want to discuss two differences when you're talking about transcription in bacteria versus transcription in eukaryotic cells. So one difference is that in bacteria you only have one type of RNA polymerase, whereas in eukaryotic cells you have three. And in terms of eukaryotic cells, the one that produces mRNA is RNA polymerase 2. So you don't need to remember that detail. But RNA polymerase 2 is the one that's used to make mRNA. Okay, so in addition to that difference, the second difference that I want you to know is how RNA polymerase binds to the promoter. Okay, so how RNA polymerase becomes bound to the promoter is different when you compare bacteria to eukaryotes. So going back to the previous slide, this is showing you a bacteria. This is showing you bacteria. And in bacteria, RNA polymerase have this detachable subunit, so that means that it can unattach from RNA polymerase. And that detachable subunit is called sigma factor. And sigma factor recognizes the promoter, and that's how RNA polymerase binds to the promoter in bacteria. So RNA polymerase binds the promoter through sigma factor in bacteria. In eukaryotic cells, rather than having one factor, you actually have a set of factors that are called general transcription factors. And the reason that they're called general, because there's also specific transcription factors for each gene. The reason that they're called general is because they assemble at every promoter that RNA polymerase 2 uses. So they're general in that sense, that every promoter that RNA polymerase 2 uses, these general transcription factors assemble. And this is what mediates the binding of RNA polymerase to the promoter in eukaryotic cells. So rather than having a single factor, you have a set of general transcription factors. And that's what they're showing you here. You don't need to remember the specific ones, but TF2D, TF2B, TF2E, F, and H, those are all your general transcription factors. And those things have to assemble, those things mediate the binding of RNA polymerase to the promoter in a eukaryotic cell. Okay, so those are the two differences that I want you to remember. There's also a difference in the termination stage, which I'm not going to go into, but the termination stage is also slightly different when you're talking about eukaryotic cells and you compare them to bacterial cells. Okay. Okay, so what I want to do is we have 10 more minutes, so I want to start by explaining something that was presented to you in one of the pre-lectures. And then what I want to do again in lab on Wednesday is you're going to need to bring your notes because we'll do a half-hour of lecture and lab again. So make sure that for lab on Wednesday that you bring your lecture notes with you, and we're going to continue on with this topic. I don't know if we'll finish it in a half hour, but we're going to continue on with it. So this picture is comparing gene expression in a eukaryotic cell on the left with gene expression in a prokaryotic cell on the right, or bacterial cell. It's also showing you what a typical gene looks like, which is in orange in the picture, what a typical gene looks like in a bacterial cell versus a eukaryotic cell. Okay, so you're looking at the DNA, and the segment of DNA that's the gene is orange. And you can see that in the eukaryotic cell, the gene has dark orange and light orange, whereas in the prokaryotic or bacterial cell, you have just dark orange. Okay? And in the picture, the dark orange regions are the regions that actually code for the protein. So those are the regions that actually determine the amino acid sequence of the protein. And so you can see that in a bacterial cell, a gene typically consists of one long coding region of DNA. Okay, so you have one continuous coding region of DNA, and that's your gene. And for eukaryotes, typically what you have is the coding regions are interrupted by these regions that do not code for the protein. Okay? And so when you're talking about a eukaryotic gene, you have both exons and introns. Okay? And exons are the names of the segments that code for the protein. And you can think of exons as the expressed sequences. And introns are the sequences that do not code for the protein. And you can think of introns as being intervening sequences that you need to get rid of. Okay, so exons are your expressed, which are the segments that code for the protein, and introns are these intervening sequences that do not code for the protein. And so for a typical eukaryotic gene, your coding regions are interrupted by non-coding regions. So your exons are interrupted by the introns. And so one thing in a eukaryotic cell that happens in transcription is that you actually transcribe both the exons and the introns. And so you initially produce this RNA molecule that contains both coding and non-coding regions. And so we said that in eukaryotic cells, one thing that has to happen before translation is RNA processing. And so one thing that has to happen in RNA processing is you have to remove these yellow introns. So once you make the RNA molecule in this picture, the blue segments are your exons, and the yellow segments are your introns. And so one thing that has to happen in RNA processing is you have to cut out the introns and paste together the exons. Okay? And that's what splicing is. Okay, so when you look at this picture, and in the middle there it says RNA splicing, that's where you're cutting out the introns and pasting together the exons. And then there's two other parts of mRNA processing that you're going to have to remember. One, you're going to have to add on something called a 5' cap, and then you're also going to add on a poly-A tail. Okay, so three things that happen during RNA processing that you have to remember for this class is the addition of a 5' cap, RNA splicing, and the addition of a poly-A tail. And all of this is happening in the nucleus, and all of those are a part of mRNA processing. Okay, so what we're going to say is that you're going to start off with something called pre-mRNA. I'll write this on the board on Wednesday. You're going to start off with pre-mRNA, and after these three processes, what you produce you can call mature mRNA. And then what you export out of the nucleus is the mature mRNA, and the mature mRNA is exported out of the nucleus, and then once the RNA molecule is in the cytoplasm, then it can be translated to produce a protein. Okay? So where we'll start on Wednesday is with RNA processing, where we're going to talk about specifically what happens for each of these processes, which essentially means you have to learn what a 5' cap is, and that's added to the 5' end, what a poly-A tail is, and that it's added to the 3' end. And I've already described splicing, and I'll describe it again, and then also add on top of that the name of the complex that performs it, which is hard to remember. It's called a spliceosome. So that's going to be the complex that actually performs the splicing process. So we'll talk about all those things. That's where we'll start on Wednesday. And then we'll probably also at least get part of the way through translation on Wednesday also. Okay, and then also on Wednesday, remember that you have two things. You have the pre-lab, and then your second stats analysis is due on Wednesday as well.